Graham's given me this title, Hard Science 101. And of course, it's it's an interesting title to have. I'm trying to make things easier for you to understand and not more difficult. So please don't be put off by the hard part of this. And the 101, for those of you who are not familiar with the American university system, the 101 series of talks is what the undergraduates get as soon as they arrive in an American university. And it's supposed to get you from knowing absolutely nothing at all up to 100 miles an hour, being an absolute expert in about 25 days. And all of the courses in American universities have these 101 courses. So today I'm going to try and get you from possibly no knowledge at all about how we do science with Aspergillus to 100 miles an hour in about 25 minutes. And we'll see how that works out. But you might need to buckle up. Okay. When I talk to patients and members of the care team and the clinicians, and usually that's in Manchester, we're based in Manchester, the questions that come up are not really the hard science questions. The questions that people ask me are, how do we treat these diseases? What do the drugs do? People are very aware of the drugs they're taking. They're aware that the drugs can be really quite difficult to take, and we're talking about drugs like azoles, and amphotericin and the candins and flucytidine and so on. And then the question obviously is, how do we find better, nicer antifungal drugs and sort of new drugs that work better for us? No problems with resistance and so on and so forth. And the next question that is often asked is, why have I got a fungal disease and sort of not everybody else? Why hasn't everybody on the road got fungal disease? Why is it just me? Fungi are everywhere in our environments. We're constantly breathing them in. Everybody's exposed in the world all the time. And you hear a talk from Animesh in India about the problems of aspergillosis in India coming up next. And this is an area that we've done considerable research on in Manchester as well. When we think about disease, we tend to start thinking about bacterial infections. This is what we're all used to learning about, and this is what most of us know about. Bacteria are really very simple organisms in certain respects. If you look on the top left there, you can see that bacteria is basically a box with some proteins and DNA in it, and sometimes they've got this fancy curly rotating tail on them as well. But there's not much complexity in the structure of a bacterium. And when you come down to it, they've got about the same number of working parts as a washing machine. That's about 2,000 to 4,000 kind of moving or working parts. So it's complex enough, but most of our understanding of disease is based on these organisms, and they're not as complex, for example, as fungi. So the fungi represented here at the bottom left of the slide, you can see that's really quite a complex thing in there. There's lots of things in there, in that kind of tube, which is the fungus, the sort of thing that grows on your jam or you see growing out in the wild. It grows as tubes, but you see there's a lot of things happening in there. There's lots of fibrils, there's lots of structures, there's lots of gaps and vacuoles and so on and so forth. So we tend to compare a fungus to something like a relatively simple car. And there's about 10,000 working parts in a fungus. And when we're thinking about developing drugs, what we're thinking about is stopping any of these parts working and hopefully stopping the mechanism working. So if you were to glue some parts of the engine in the car together, you might stop the engine working. The car wouldn't go anymore, and that would be a way of stopping the car working. In the same way, we have drugs which interfere with components of the fungus, and they will interfere with a system in the fungus, and they will stop the fungus working. When we look at drugs and when we think about drugs, that's what we're thinking about. We're thinking about something which stops the function of the mechanism. So we've got about 10,000 parts to play with in a fungus. Of course, when we take a drug, we're not just giving it to the fungus, we're giving it to a human being. So when we move to human cells, they're a little bit more complicated still. You can see there's a bit more inside the human cell. And of course, human cells are all connected together to make flesh and bone and body. So there's about 25,000 parts, comparable still to a more complicated car, but quite a few more working parts. But when you think about the bacteria and the analogy to the washing machine, a lot of those parts are not the same. Whereas you think about the fungus and you think about the human, a lot of the parts are shared. They both have engines, they both have wheels, they both have brakes, so on and so forth. So fungi are very closely related to humans, and bacteria aren't. So they share a lot of parts. So making a drug that works against a simple car, but doesn't work against a complicated car is really quite difficult. You've got to choose something that the complicated car has that the simple car doesn't. And that's where the problem in developing drugs comes in. So how do we find antifungal drugs? The first way, and the traditional way, is to do a high throughput screen. We take thousands and thousands of different chemical compounds, and we just add them to fungus growing in these little dishes with lots of little wells in them, and we see which wells the fungus doesn't grow in. And you can see on the right here that there's a couple of promising candidates here where the fungus doesn't grow very much at all. So that's a very simple brute force method of discovering compounds that kill the fungus. And we screen hundreds of thousands and hundreds of thousands of these compounds to try and find something that works well. An alternative method, and one that we're developing in Manchester, is to go back to the fungus to knock out the instructions that code for the parts of the fungus and see which of those instructions is important. So we don't use drugs here. We're simply removing the instructions that make each part of the machine. And then we're seeing, well, this particular part of the fungus, this particular part of the machine, it's really important to make it work. The fungus is dead. The fungus doesn't grow very well. So we go back to our car analogy. Basically, what we're doing is crashing the car a lot. We're taking things away and seeing if it breaks. And mostly, when you take a part of a car away, you take away the floor mats, you take away the back seats, you take away the boot, the car will work perfectly well. Maybe not quite as well, but generally speaking, you won't notice the difference. So most compounds and most of the instruction sets we take out don't do anything whatsoever. And in the bottom, you know, we screen and screen and screen and remove these instruction sets, these genes, and nothing happens. So the majority of our time is spent discovering nothing. But every now and then, on the right side of the screen, we discover something that stops the brakes working. We discover that hitting a car with a tree will stop it more reliably than hitting it with a feather, and so on and so forth. So we discover ways that components of the fungus are needed, and we can call these targets for drugs. So I'm going to talk a little bit about the azoles. I know azoles aren't the only drug that we use against fungi, but I'll talk about how the azoles work. It's very common, and I think a lot of people who are treated with antifungals will be familiar with itraconazole, voriconazole, fluconazole, posaconazole, and so on. Now, on the left here at the top, this is a component of the fungus. This kind of amazing structure made of coils and wires, it's red coils and green wires, is one of the components of the fungus. It's a protein. And it's involved in converting this compound lanosterol ultimately into a compound called ergosterol. And the fungus needs ergosterol. So if you stop this process, you will kill the fungus. So if you look on our top left figure there, you can see that lanosterol, that's the chemical compound that's being acted on by this amazing little machine, is shown here in blue, and it fits inside this protein, this structure, and that's how the protein changes it and moves it along the synthesis chain towards ergosterol. If we look at our antifungal drugs, and here we're looking at itraconazole and voriconazole, you'll see that they fit into the same place that the lanosterol should fit into, and they get stuck there. And they don't come out, so it stops the lanosterol getting in, it stops the action of this particular protein. No ergosterol is made, and the fungus will die. And you can see that itraconazole is sat in there, it's quite a big molecule, and voriconazole, which is the sort of darker blue, is also lodged in that exact active site. It's a much smaller molecule. So that's how drugs work, and that's what we're aiming for when we do a drug screen. We're aiming for a drug which sticks to an important component of the cell and stops it working. The next question people say is, well, yeah, great, we're taking azoles. Why do I feel terrible? And the reason is that the human cell and the fungal cell are actually quite similar. So if you go back to our analogy of the car, they have the same systems. They both have, for example, engines or brakes or wheels. So trying to find something that works only on the fungus but not on humans is actually quite difficult. In the case of the azoles, again, if we look at alanosterol and itraconazole, humans actually also have this component in quite a similar form, not exactly the same, but it's a bit similar. And it's similar enough for the azole drugs to stick and bind a little bit to this human structure. Now, in humans, this pathway and these structures go on to make cholesterol, and cholesterol goes on to make steroid hormones. And the humans actually have a variety of other types of protein which are very similar to this as well. And the azoles bind to all of these. They don't bind very well. You know, they really are designed quite carefully to bind to fungal components, but they do also a little bit bind to the human components. And if they do that and they inhibit, you'll get less cholesterol and less steroid hormones and less activity in your liver of these types of proteins that do detoxifying and other important functions in the cell. And that we call off-target effects. And that really is why you get these side effects. You'll find that, you know, as we've gone over the years, moving from itraconazole for Aspergillus to voriconazole and isavuconazole and the newer compounds, that there are less and less of these kind of side effects. And that's because they are either much more active against the fungus and you need much less of them, or they bind much less to the human components that are similar to the fungal components. Okay. So following our drug discovery methods, and in this case, it was a mixture of the two drug discovery methods, screening lots of compounds and actually carefully removing the instruction code of the fungal cell. There's a new drug just coming in, or just on its way through phase 3 clinical trial, so it's quite close to being in the clinic, called olorifim. And this drug fits into this protein. I'm not going to tell you what it is, but humans don't have the same protein. They have something which does more or less the same job, but they really don't have the same protein. So this drug has the potential to be much, much better in terms of treating patients without side effects. This has been developed by a company called F2G Limited. They're in Manchester. And I think Ange is going to go on and tell you a little bit later about all the new drugs that are coming through the pipelines, and it's really quite an optimistic picture going forwards. And we've got lots of these drugs appearing. So that's the drugs. That's what we're doing in our labs in Manchester, is we're looking at ways to find new drugs. We have hundreds of possible targets, and it's really, for us, it's a matter of narrowing down and finding the right ones to move forward with. Okay. The next question we're addressing is kind of, why do I have fungal disease? Why, and in that sense, I mean, why me? Why have I got fungal disease and not other people? Again, we've got to think, you know, we tend to think of disease in terms of bacteria, and now, obviously after COVID, especially a viral disease. And that's what our concept of disease is. Fungal diseases are rather different, however. So with a bacterial and viral disease, as you're well aware, if you stand next to somebody who's coughing and sneezing on the bus or in a supermarket queue, they can infect you. They breathe out the virus or bacteria, you breathe it in, and you become infected. And then those people will go on to infect other people, and the disease will spread through the population. So that's what's happening on the left here. In fungal disease, however, that's not what happens at all, certainly with aspergillosis. So if one person is infected with Aspergillus, they don't infect other people. It's incredibly, incredibly rare for that to happen. So they don't infect other people. So the people who have Aspergillus and aspergillosis infections have acquired the infection from the environment and not from other people. So it's very different to what happens in the bacterial disease. So if people are acquiring the disease from the environment, and the environment is really full of fungus, so we're all exposed to it, that means that the people who don't catch it must be resistant to the disease, and the people who do catch it must be sensitive to the disease. So what the difference is there is in how the people who catch it have different sets of components, different sets of DNA than the people who don't catch it. So the question you've got to ask is not, why do I get it? The question is, are you and Aspergillus compatible? So everybody, like every car, there's a blueprint. So the blueprint tells an engineer which components to make and how to put them together. And the end result is a working machine. It's the same with a human cell and the human body. Our blueprint is DNA, and the DNA tells the cell what proteins to make and how to assemble them and into a working cell. And then it tells the cells how to assemble themselves into a tissue and a working body. So everything's from the DNA, and the DNA is the blueprint. So if we're looking for differences that make you susceptible or resistant to Aspergillus disease, we look in the DNA. We've known for years that people who have Aspergillosis have a very different immune system to people who don't have Aspergillosis. And don't worry about the details of this diagram, but these are all immune molecules, and they behave very differently in people, in this case, in people with chronic pulmonary aspergillosis than in healthy individuals, when both of these are challenged with Aspergillus fumigatus cells. And you can see the differences are very big indeed. And if you look at the graphs on the left, you can see on the right side, some of the responses are much, much bigger or much, much smaller when you have susceptibility to Aspergillosis. And we've known what the responses are. We've known the differences in how the immune cells communicate to each other, and these responses, we've known about them for really a long time, but we've never known why these responses are different. What's the fundamental cause? And that fundamental cause is in the blueprint, it's in the DNA. So the way we address this as scientists is we look at a population of healthy people, and we look at a population of people with the disease, and they're in red in this case. And we compare DNA from people with the disease and people who don't have the disease. And we sequence all of the DNA and we look for differences. And what you'd expect, of course, is that there's going to be differences shared by the people with the disease that just aren't going to be there in the people who don't have the disease. So we did this, we've done this with ABPA, and we've done this with CPA as well. And we find, yes, there are genetic differences between the people who have diseases and people who don't. And our first study, we started this in about 2010. It takes a very long time. There's a lot of DNA in the human cells, about 3 billion bases, and it only takes one difference to make you susceptible to disease. So that's 1 in 3 billion. So it's a very difficult process to screen through all this information, sift through it, and find the results. So with ABPA, we were looking for differences that, were present in people with ABPA, but that weren't in people who just have asthma, for example, or the healthy population who don't have any diseases. And we didn't really find any. What we found is that we have this variant here, and this is one of the most important differences we found. So this is a single base in the genome of individuals. And it's quite often in ABPA, but we found it was also very often in the genome of people who didn't have ABPA. And we were a bit confused about this. But when we looked, we noticed that people who had this variant, and this single change in the DNA makes a big change to the gene and the coding instructions. It's like removing the NT from do or don't in an instruction. We found that the people who have this variant have 15-fold more fungus in their lungs than people who don't have the variant. The variant's only present in 4% of people in the whole population, so it's quite rare. But it's present in 60% of people who have ABPA, and people who have ABPA who have a lot of Aspergillus fumigatus in their lungs, specifically Aspergillus fumigatus, not other fungi, which is one of the reasons we were having difficulty with this particular variant. So this is a variant that means you get more fungus in your lung. And this is to do with what the cells produce. They produce a sticky matrix that helps them stick together, on the surface of your lung. These cells that have the ZNF77 produce a lot more of this. The fungus likes this. It grows a lot faster in the lung. It gets the germination stage about 2 hours more quickly than in a healthy lung. And that completely changes the amount of fungus in your lung because it grows in a three-dimensional manner. If it's growing twice as fast, you can imagine that means you get a lot more fungus. And it changes the way as well that the cells signal to each other. And these are the changes that we've seen previously in the immune systems of patients. So this is one variant that seems to be very important partly in ABPA, but also in determining whether people are susceptible to fungus. This, by the way, is specific to Aspergillus fumigatus, this effect of increasing the speed of germination. It doesn't work with Aspergillus flavus, Aspergillus niger, other members of the Aspergillus species. It doesn't work with other fungi. So this is specific to Aspergillus flavus. The second thing is that this is a gene that's not present in any other animals. It's only present in primates, the most highly evolved monkey type group. So it's not there in mice, it's not there in rats, it's not there in most of the animals we use for our model testing systems of disease. So this is a variant that we would never have found using animal models of disease because the gene isn't there. So it's only because we've been able to work with patients and work with the DNA and the genomes of patients that we've been able to find this particular variant. In CPA, this is an ongoing story, and again, we started this about 12 years ago. So we have patients with CPA and patients who don't have CPA, and we look at the changes in the genome, and we're just beginning to understand some of these changes. We think we found a particular change in a gene called ITGB3, and ITGB3 is right at the top of all of these signaling processes that govern repair, proliferation, and inflammation in the human lung. And these are the processes, again, these are the changes that we see in the cells when we look at them. And this looks like it might be a very good candidate for something that controls this, sort of the root cause of what makes you susceptible to CPA. So we're very excited about this. We haven't finished working with this and we haven't finished proving it. So it's very early days yet, but we're very excited about the possibility that we can find the master variants that govern susceptibility to fungal disease. And we're excited about that because it might be really good to screen people proactively. For example, people who've had tuberculosis or sarcoidosis or COPD, it might be very good to screen them because these are the people who get chronic pulmonary aspergillosis in the main. Be good to screen them and find out which of them might be susceptible to aspergillosis so we can take some action earlier, either just by looking at their lungs more often and being very, very proactive in monitoring, or perhaps by treating them earlier as well. So we're very excited about finding the genetic basis of these diseases. The work's being carried out in the Manchester Fungal Infection Group. We're one of many research groups around the world. You're going to hear from Animesh next in New Delhi. And there are hundreds, thousands of researchers doing the same sort of work around the world. So we're part of a large global community doing this. And it's the large scientific effort that's now coming into play. It's not always been so, and Graham mentioned that 20 years ago things were rather different. But now there's a very large global effort aimed at working with aspergillosis, understanding how it works, and producing new diagnostics and treatments. Particularly, you may recognize some of the faces at the bottom there, but David Denning, Mike Bromley, Sarah, Amarga, Malcolm Richardson, Takanori, Rena Richardson, and some absent friends have been involved in this work as well, and it wouldn't have been possible without them. It's been funded by a wide range of organizations. Fungal Infection Trust, National Aspergillosis Centre has helped massively, MFT, EU Framework 7, MRC, Northwest Lung Centre Charity, Gilead often fund us of small grants, and so on and so forth, and other industries. F2G support us a lot. So we get lots of input from a lot of people. So I'm going to finish there. I mean, Ange is going to tell you about some of the exciting new developments in more detail. Animesh will I think give you a really interesting insight into what's happening globally. But I'm going to leave you there, and I'll take questions, or I can take questions at the end.
Paul, we've just got one question in the chat, and
Okay.
it's from one of our patients, and she just asks, why do the azoles stop being effective for some people?
Yeah. So when you think about, I'll go back. If I can go back, yeah. Yeah, okay. With azoles, there's a couple of things that go on. You can see that the azole molecule fits really nicely into its target molecule. Really precise fit. And that's the only reason it can work so well, is that the molecule, the azole molecule, you can see on the left here, itraconazole in light blue, voriconazole in dark blue at the bottom there, they fit really precisely into that pocket. Now, because this is this component of the fungus is a protein, and it's coded for by a piece of DNA, if that piece of DNA changes, it can change the structure or the way this protein folds itself up. And if the protein folds itself up slightly differently, you can imagine it can stop the itraconazole or the voriconazole getting in. And that means that the drugs really won't work as well anymore. So the fungus can mutate and evolve. And sometimes that's what happens, and it stops the azole working so well. Obviously, it's got to keep having a function as an enzyme to work on the lanosterol. So the mutations tend to be pretty specific and not very common, but that's what can happen. So you can get a mutation in the gene that codes for this protein that can change the protein structure, and it stops the drug fitting so snugly into that pocket. So it doesn't work so well. And that's, you know, the fungus can develop resistance in that way.
That's great. Thank you, Paul. We've just had one more drop into the chat as well. And this is from Johanna. And they say, are there no orthologs of ZNF77 in other animals, or do they not produce any similar proteins?
Yeah, it's there in gorillas and some of the apes. It's not there in anything else. So it's unique to humans. And if you go back to the beginning of the talk and you think that fungi have got 10,000 components, 10,000 genes, that means humans have 25,000, and other animals have less genes or more genes, but they have different sets of genes. So it's not the case that every gene is present in every organism. And this one, as I say, it's quite interesting that it's only present in humans or the higher primates. And it's quite interesting that the effect of it is specific to Aspergillus fumigatus. So to me, it seems like there's been some kind of evolutionary event here where the fungus has evolved to take advantage of this. And perhaps some of these other animal species have lost the gene because of sensitivity to fungal attack, or they've mutated the gene or changed the gene, so it's not there anymore. So it's quite an interesting evolutionary question. Where did this come from? And you know, why don't any other animals have it? We don't really know, is the answer. But again, if you think about a mouse, the lungs are really, really tiny, and the airways are really, really tiny. So if a mouse were to produce all these sticky compounds in the way that human lungs do, they'd probably just clog up. So it's probably not, you know, it's not going to work for a mouse to have this kind of gene, right? Whereas human lungs are nice and big, and you know they can stand having all of this sticky mucilage around and these sticky compounds around. So maybe that's the basis of why animals don't have this. We don't know is the answer, but we can speculate.
That's brilliant. Thank you, Paul. We've got no other questions in the chat.
I can't resist asking one question. Having told us you found the root gene for CPA.
Maybe.
Maybe. So you're going to go and look for it. That's one suggested way to help to prevent people getting aspergillosis. But what most patients will want to know is that if this proves to be one of the root causes of why people are vulnerable, does that suggest ways that people could be treated better?
I think so. I mean, if this does turn out to be a root cause, it's going to be one of several. Is the first point to make. I don't think that, you know, the susceptibility to the disease is that simple. It'd be nice if it was, and it would be great if it was. But there are drugs that work on these compounds already, because ITGB3 is involved in many other different types of disease. It's kind of a master regulator. So we already have drugs that work against it. It would be interesting to see if they work in the context of aspergillosis. And yes, it would be a good target to develop new drugs.
That's really good news. Right. Thank you, Paul. Excellent talk as always, and a nice little nugget of excitement at the end there.